So good afternoon, everyone. My name is John Clark, and I'm hosting today's colloquium. To present our colloquium, I am pleased to introduce Professor Lydia Sohn from our Department of Mechanical Engineering. Lydia received her bachelor's and master's degrees from Harvard and subsequently her PhD there in 1992. Her thesis advisor was Professor Mike Tinkham, and Mike and I knew each other very well and collaborated over a number of years. And during one of my visits to his lab, I first met Lydia. And subsequently out of that research, she and I jointly wrote a physical review letters with some other authors. After her PhD, she became a postdoc, first at Delft University of Technology in the Netherlands, and then at Bell Labs, until joining the Princeton faculty. She moved here to Berkeley in 2003. So from 2011 until now, she has been faculty assistant to the Vice Chancellor of Research. This fall, she became Vice Chair of Diversity, Equity, and Inclusion in the Mechanical Engineering Department. And last year, she became the Almini C. Maynard and Agnes Ofield Maynard Chair in Mechanical Engineering. Lydia's graduate research at Harvard and also subsequently at Delft were focused on the fabrication and measurement of superconducting Josephson arrays, and a field, of course, is very close to my own over the last years. And since 2000, she has applied her technology to develop label-free methods to investigate biological systems. Now, that's been very successful, and in recognition of her achievements in this field, in 2017, she was elected a Fellow of the American Institute for Medical and Biological Engineering. So we're going to learn about both technologies during her talk today, and as you see on the slide, the title is Node-Pore Sensing, how a humble four-terminal measurement can measure the mechanical properties of single cells. And incidentally, if you have questions, please post them in chat and I will read them out after her talk. So please, let's welcome Professor Sohn to give her colloquium.
Well, thanks very much, John, for the very kind introduction. And thank you very much for inviting me to speak about some of the work that we've been doing in my lab. I want to just sort of, since I'll be talking primarily on Node-Pore Sensing, which is a hallmark technology in my lab, I thought I would just give a little sense of other things that we're working on in my lab. Let me see, why aren't I able to? Oh, interesting. Oh, okay, here we go. Sorry, so overall, we're developing platform technologies wherein we can actually study single cells and see how they react and interact with their environment. And one of the technologies that's becoming fairly advanced in my lab, and I just thought I'd give you just a brief introduction, perhaps you can find it of interest in your own research, is that we have now developed a way that we call high-throughput DNA-directed patterning. We actually use traditional photolithography, the same technologies that I've used to make Josephson junctions, to create photoresist templates that we can then use to pattern different single-stranded oligos of different sequences on a glass chip. And we can pattern and make new templates, strip off the resist and so forth, and continue to do our lithography. The amazing part is that whatever oligos that we pattern onto our substrate, they don't get damaged through that very harsh process of doing lithography, of stripping with acetone, using a very basic or alkaline developer. And what you see here is how well we're able to pattern different oligos. We've taken oligos that have been attached to a different fluorophore, a different color fluorophore, just to show people exactly what our precision is and how well we're able to do this. Now, what we do with our DNA patterning is that we can actually pattern different cells. Each cell is tagged with an oligo of a particular sequence, and we can wash these cells that have been tagged with the oligos onto our pattern glass slide, and through Watson and Crick hybridization, the cells land exactly where we want them to be. So here's just the canonical Berkeley logo, just showing how well we can do this. We can also pattern different proteins and antibodies onto our substrate. And this is just an example that these are different culture conditions that we've created onto our substrate so that we can look at what happens with single cells as they interact with their environment. And this is just an example of cells, neural stem cells, as they're feeling their different environments. Teal corresponds to one protein, purple corresponds to a different protein. And you can see the cells are very active feeling their environment, which was really neat for us to see over a time course of 5 days. We're also using the same technology to look at cancer dormancy. And really, this is my postdoc's project of really why does prostate cancer, men who have prostate cancer, they will go into remission and then years later suddenly develop into full metastatic disease. And she's looking and recapitulating the bone microenvironment. And if you can see, there's this little tiny green dot right here. That's actually a single prostate cancer cell that she's patterned into her bone microenvironment. So we have this very extreme, nice way to precisely pattern cells. We've also used this same technology to look at exosomes, which are small packets of information that cells use to communicate with each other. And also we had our hand in labeling virus with oligos so that we can actually do community surveillance, looking, for instance, for the SARS-CoV-2 virus in pools of samples. But today, that's just an overview, but today what I really will be talking about is mechanophenotyping with a device that we call Mechano-Node-Pore Sensing, or Mechano-NPS. But again, I just wanted to give you a flavor of the things that we're able to do in our lab. We build essentially platforms that then we use to investigate biological systems. So what I'm going to be talking today is really how we use mechanophenotyping, use this very simple device where we measure current using just DC voltage to look at breast cancer cells or potential of having breast cancer. And I thought it was fitting that it's October, and October is, as many of you may know, is the Breast Cancer Awareness Month. And just to give you some statistics about breast cancer, I was actually shocked to find out this year in 2021, breast cancer is now the most commonly diagnosed cancer worldwide. It used to be for a very long time lung cancer, but now breast cancer has overtaken that unfortunate position. About 1 in 8 women in the US will develop breast cancer in her lifetime. And if you look at the overall 5-year survival rate, it's not bad. It's 90.3%. But of course, that depends upon the stage at which the breast cancer was diagnosed. And as you can imagine, the later stages, the survival rate goes way down. Ultimately, about 43,600 women are projected to die this year, breast cancer, and the majority of that is due to metastatic breast cancer. In fact, metastatic disease accounts for 90% of all cancer deaths, no matter the cancer. Now, if you look at breast cancer diagnosis and age, this is from the NCI SEER epidemiology study. And what you can see is that as a woman progresses in age, there is more likelihood percent of new cases of breast cancer, typically between 55 to 74. That's when the majority of breast cancer is diagnosed, and then it falls off. And because of this sort of later stage of life where breast cancer is diagnosed, breast cancer is a disease of aging. Now, given that, at the same time, there are risk factors that do complicate things. For instance, you could have a BRCA1, BRCA2 mutations, which hopefully many, I believe, has been in the news quite a bit. But essentially, these mutations increase the risk of breast cancer in a woman, and because of these mutations, the breast cancer could be diagnosed form at an earlier age. So, of course, the question is, what are their methods? What methods could there be for early surveillance or diagnosis? Now, one of the things that we got interested about 5 years ago in my lab is something called random periareolar fine needle aspiration, or RPFNA. And it's been its moniker is known as the Pap Test of the breast. And what that is that for women who already have a high risk of disease, what a doctor could do is random sample the breast tissue with the understanding that if breast cancer, if tumors are starting to develop, changes of the breast occur, it's not just a local event, it actually affects globally around the breast. So if you randomly sample, could you actually, in the end, capture some of these cells or see cells that are transforming into something that's, cause for concern? Now, with RPFNA, women would go in, for instance, once every quarter of a year to get this done, and cells are randomly taken. They are then screened using various types of immunochemistry to highlight different proteins that may say that the cell is a rogue cell. The problem is that this is done under a microscope, and it's really just done by a pathologist. So it's a qualitative investigation or imaging of the cells. The problem is that some women come in to the doctor's office, there's no sign of any disease, but the next time she appears, suddenly she has metastatic disease, and there was no indication months before. So clearly, this qualitative assessment of cells from this biopsy is insufficient. And so what we wanted to know is whether or not, is there another way to assess these cells quantitatively? And that's the important thing, is quantitatively. Could we actually capture some of the cells that are transforming into potentially a dangerous cell? So what I hope to convince you is that you can do mechano-phenotyping of cells, and that actually provides a lot of information about the cell that is complementary to traditional biological assays. And so sort of the hypothesis or the question that we asked in our lab, sort of focusing on RPFNAs, was whether or not could cell mechanical properties in the end be used as sensors of biological states or future outcomes in cancer. Now that's a mouthful, but what I hope to show you is that it's really amazing how much information you can just from squishing a cell, essentially, and that's what we do. So I just want to give you an overview of the different technologies that people use to actually mechanophenotype, to assess the mechanical properties of cells. So the gold standards are micropipette aspiration and optical tweezers. And you can get exquisite information about the cortical tension, the Young's modulus of these cells, and so forth. The problem is that these methods are very slow. With micropipette aspiration, it could be just a few cells in a day in terms of being able to assess their mechanical properties. The AFM is also a gold standard, and that one you get a lot of information, viscoelastic properties, but again, very slow, just a couple of cells in a day, which is rather astounding. Optical tweezers, microplate rheometry, and microbead rheometry, all those again give you quite a bit of information, but they have very low throughput. Recently, there's work on developing something called microfluidic cytometry, where you basically have a cross slot of flows, and essentially you deform the cell and look at its mechanical properties. With that, you can achieve very high throughput, but one would question about really how much more information could you get compared to, for instance, an AFM or micropipette aspiration. You can get quite a bit, but how competitive it is. So we did a sort of global search on all these different methods, and what we realized is that if you consider throughput and resolution about the amount of information that you can get, there is quite a bit of room for new methods. That is something that would have high throughput, but also have high resolution, meaning getting as much information as you can about the mechanical properties of cells. So with that sort of introduction, what I hope to show you is this method that we call Mechano-Node-Pore Sensing really allows you to achieve this sort of relatively high throughput and also quite a bit of mechanical information or resolution. And I really want to emphasize again, this is, as I mentioned at the very beginning, and it's in the title of my talk, this is a very simple 4-terminal measurement to mechanophenotype cells, which kind of sounds strange, right? Because you're measuring current and voltage, but you're measuring something mechanical. So I always sort of like to emphasize this is like a very different measurement. I should say, with the other techniques that I've highlighted, all of those techniques use a high-speed camera, and it becomes very costly. Okay, so let me sort of back up and explain to you how we got here in terms of Mechano-Node-Pore Sensing. So it really started quite a while back, really in 2000 or so, when essentially I moved over from Josephson junctions over to biology. And one of the things that we did in my lab was something called resistive pulse sensing for screening cells. And for those of you in the audience, you may know this as the Coulter counter principle. It's a very simple technique. And here's a picture, an image of an actual device that we made in my clean room. And what you can see is that we have a primary channel, and I'll interchange that with pore, and that's just because of the sort of how we started out on this project. It's historical reasons. But what you can see is the channel bridges across 2 different reservoirs with filters. Okay. What you also see, for those of you who have done 4-terminal measurements, we've put down 4 electrodes where we've separated out the current and the voltage. Okay, and you can see the dimensions of our device. Now, the filters are just to filter out any stuff that may be in our cell solution, but what we do is we run cells across from one reservoir to the other through this channel. And when we do that and we apply a current, we can measure the voltage or measure the resistance. And here what you see is essentially what happens. This is current versus time. And then as a cell passes through this channel or pore, what you see is we have measured some constant current when there's no cell in the channel. And then we suddenly see as the cell drops off into the channel, we see a drop off in current until the cell all the way passes through into the next reservoir, and we see a rise in current back to baseline. Now it turns out that the height of this pulse, or the magnitude of the current pulse caused by a cell passing through the channel, corresponds to cell size. And the way to figure this out was been done in the early 90s, and whereas really what you have is, in the end, I'd like to emphasize that what is a microfluidic channel once it's filled up with water? It's nothing more than essentially a wire, a conducting wire. So you have electric field lines, you have current passing through your from one reservoir to the other. And when a cell passes through, that cell is essentially an insulator, it's a dielectric. And what it's going to do is it's going to block off electric field lines, it's going to squeeze and disrupt the electric field lines. And it turns out that it was Gregg and Steidley and Dubois and Bean who actually calculated what the change in resistance that you would measure as a particle would pass through, depending upon whether or not that particle was comparable to the size of the diameter of your channel or whether it was much smaller than the diameter of the channel. And you can see that these are nonlinear equations. They were solved, with Maxwell's equations and so forth. The main thing that you should glean out of this is that the change in resistance that you measure is really dependent upon the size of your, the length of your channel, the diameter of your channel, and also the particle size. And this F factor right here, I usually joke around, it's really a fudge factor, but that was just determined numerically by doing many experiments. But in the end, despite how nonlinear and how ugly these equations are, ultimately, it's really nice that you can always estimate the change in current that you measure in terms of it's really approximately a ratio of the volume of the cell with respect to the volume of the channel of your device. So if you go back here, again, the larger the cell is comparable to the size of the channel, you can also think of it as displacing conducting fluid, or it's blocking current field lines. And that's the reason why you get a drop off in current. The larger the cell, the more the height changes or the magnitude of the current pulse changes. You can also look at the pulse width or the duration of the pulse, and that corresponds to transit time. How long did it take for the cell to pass through? And we've taken advantage of that to look at different proteins that are on the surface of the cell, whereby what we do is we take antibodies that would correspond or bind to a specific protein on the surface of the cell. We take antibodies, we functionalize the glass slide that this channel, which is made out of PDMS, is bound to. And if the cells can transiently bind, then the cells will take much longer to pass through if they have the protein that interacts with that antibody very specifically. And you can see that in terms of a transit time. And we've used that to screen different cells for particular markers that are on their surface. It turned out and this is this device that I think I always tell people it's a project and a device that keeps on giving. It keeps on morphing into something because we discover something new. And as I mentioned, this happened about, I would say about 10 years ago, when again, this whole idea that what is a microfluidic channel when you measure current? It's really nothing more than a wire. So here's a current pulse that we measured when a cell passed through this particular device. But then what we could do is we can actually widen parts of this channel, and we actually call these widened areas, we call them nodes. So here's a pore or channel, and here's a node. And essentially, again, think of it as a wire. We're changing the current density. So a cell passing through, suddenly the current pulse goes from just a square pulse into something that's bimodal, like that. And what is the reason for this? Again, if you have conceptually this idea that the current that you measure is really a volume to volume ratio of the cell, or another way is it's displacing conducting fluid, so you're going to see a drop off in current. When the cell is in this narrow portion of the device, you're going to see a drop off in current. But then when it enters into a node, that node, there's much more conducting fluid. So the effect of the cell passing through is less, you see a rise in current. And then once the cell leaves that node, then you see a drop off in current until it leaves effectively the device. And for us, that was just really astounding. Like, wow, this is something that we can take advantage of and do different things. And so for us, what this is just showing you is that here what we did was we created a Fibonacci sequence of nodes and pores, and you can see that the current pulse reflects the nodes and pores that we have in the device. And so what that really means for us is that with Node-Pore Sensing, as we now call it, essentially we always know where a particle is in that channel. In addition, we can encode our channels with spatial and temporal information. And what we do is we get unique current pulses that come out. And that means it's really easy for us to find these pulses against a background of noise. Effectively, we have a high signal to noise ratio in that regard. And we can play around and we can use different algorithms that correspond to, for instance, Wi-Fi algorithms, cell phone tower algorithms to separate out different signals from multiple cells entering into the device, or we can multiplex. So it's a really neat and very flexible tool, this Node-Pore Sensing. Now, what we ended up doing was, and I mentioned this at lunchtime to John and Hernan, was that essentially I had this mechanical engineering graduate student, Jung-Hyun Kim, and I sent him off on a goose chase on another project. And he decided that no, he wanted to be a true mechanical engineer and he wanted to measure mechanical properties of cells. And he said he wanted to take advantage of Node-Pore Sensing to do this. He explained his reasoning, he showed me a sketch, and I was like, okay, you can give it a try, let's see what happens in a couple of months. And from that, it really just started a whole new direction for my lab. So he was just a brilliant graduate student, as you all know Berkeley students are. And what you see is his device is complicated, but at the same time, very simple. So here are his 4-terminal measurement electrodes, and he's measuring the current across this device. And you can see that we have nodes and pores. And then we have a channel that we call contraction channel, and it's very narrow. So essentially, a cell has to squeeze through to actually pass through the entire device. And then we have a series of nodes and pores that we call recovery. And I'll explain what I mean by that in just a second. But if you look at the cross-section of our device and when a cell passes through, in the Node-Pore section, the cell is just happily passing along through. It has its, we call it the free diameter. It's just suspended and buoyant in the cell media. But again, as I mentioned, the contraction channel, the channel is very narrow, so the cell has to really squeeze through to pass. Okay, there's a thin layer of fluid between the cell and the PDMS microfluidic channel, and it's enough to allow the cell to pass through. And what's the kind of current measurement that we get? This is a current pulse that we would get. And as you can see, we get the up and down current pulses that correspond to when the cell is just passing through these nodes and pores. When it goes through this contraction channel, you're going to see an extreme change in the current pulse because it's squeezing through. It's displacing a lot of conducting fluid. And so you see a drop off in current. And then finally, it returns and gets to relax back to its original shape. And with that, with this current pulse, we can get many pieces of information about this cell. We can get the diameter of the cell just by, again, looking at the current pulse magnitude in this first section right here. We can get stiffness. How long did it take to pass through that contraction channel? If it's very stiff, it's going to take a long time to pass through. So we're looking at duration of that pulse. Cell deformation, how much as the cell gets squeezed into this narrow channel, how much does it go this way? You know, it's being squeezed this way, so it's kind of deforming upwards. And cell recovery, which becomes a very big part of my story in the next series of slides. Now, just to give a little bit more information about cell recovery, I just want to explain at the beginning, before I start showing you data, we classify recovery in terms of whether or not the cell instantaneously recovers from being squished. And how do we know that? Because we see that these current pulses, after it's being compressed, return back to this initial current pulse magnitude right away. We can also have cells that we say have a transient recovery, and it takes them longer to essentially reach their original size. And again, you would see that in terms of the magnitude of the series of current pulses. When does it return back to what it was before it got compressed? And then finally, there are cells that we say just have prolonged recovery. We couldn't measure it. They just, within our resolution of our device, which what I'm showing you with data is about 40 milliseconds, they just didn't recover. The beauty about using photolithography, about using PDMS and so forth, is that we have quite a bit of design flexibility. And now we have a recovery time with some of our devices that go over 200 milliseconds overall. So what can we do? So Zhang Yang, again, to prove his point that it was worthy to pursue this, this really, in the end, fantastic project, was he just looked at cell lines. And here what he looked at were breast epithelial cells that were non-malignant versus malignant cells, MCF7s. And what you see is a three-dimensional plot of the transit time of the cells to pass through that contraction channel, the original size, or what we call the free cell diameter of the cells, and then L deform, how much did they essentially get deformed this way when they got passed through that contraction channel. And you can see there's quite a bit of overlap, but in the end, you could still see groupings between differences between these two different cell types. As well, here is just a sort of a purple gradient of essentially the recovery time. That's not part of the story yet. Here he also looked at different cell lines. This was for lung epithelial cells, non-malignant versus malignant. And again, he can see differences between the two different cell types. This was with an applied strain of 0.3. The problem is that you can see that there's still a bit of overlap. And in case of the Bs2B and A549s, that overlap was about 30% in the end. And you can think about it that even within a population of cells, some cells are going to be larger than others. And that's all going to affect the transit times. Like cells that they may be of the same stiffness, different size. A larger cell is just going to take longer, again, to pass through versus a smaller cell. So Jung reached into his bag full of tricks as a mechanical engineer, and he ended up using something called the Buckingham Pi technique, which is really a dimensional analysis method where you take in all the independent parameters and just, again, do dimensional analysis to create an index. And we now call this, for us, we call it whole cell deformability index, wherein we take in all these different parameters of our microfluidic device, and we take into account the size of the cell and how long it took to pass through. So a larger cell is going to take longer to pass through versus a smaller cell is going to take a shorter amount of time. So effectively, with this Buckingham Pi technique, what we're able to do is essentially take into account size of cell and normalize it in our measurement. And in the end, this whole cell deformability index, or the WCDI, really describes the whole cell deformability. And that means including what happens to the nucleus, all the organelles that are in the cell, and the cytoskeleton, which is really the basic architecture structure of the cell, why the cell stays in its particular form. So, but what does it really mean to have an index? Even though I'm saying I'm normalizing the size of the cell, but what does it mean physically? So we did some side-by-side comparisons using micropipette aspirations. And this was done by a postdoc in Zev Gardner's group at UCSF. And we looked at 3 different cell types. And what we found is that the whole cell deformability index, the WCDI, is inversely related to cortical tension. So a high WCDI means that a cell is soft, versus a low WCDI, it means that the cell is stiff. So you have to keep in your mind inverse of numbers. If you compare it to what's been done with AFM of these different cell types, again, AFM will measure Young's modulus, and what we find is our WCDI is inversely related to the Young's modulus. And so what you can do is, instead of a 3-dimensional plot of these different parameters, what we found was, again, we wanted to take away the possibility of cells being of different size. And what you find is by applying the WCDI, you get these really nice histograms with respect to WCDI. And you can see that the overlap between the 2 different cell types, once we've removed the size of the cell, you can see that the overlap, it went essentially from 30% down to 1 to 2%. So it was quite amazing. And again, high WCDI means that the cells are soft. So these are malignant cells, and they're softer than the non-malignant cells, which is consistent with what everyone feels and thinks and measures of cancer cells. They're softer generally. Okay, again, we can look at the recovery times, and we see variations. Not part of the story yet. In terms of could this method actually measure when you actually disrupt the cytoskeleton? And the answer is yes. We can apply LAT-A, which basically obliterates the F-actin network in cells. And what we see is that when we do that, we see slight changes in terms of the stiffness of the cells become softer. Again, there you see an increase in WCDI, so that means the cells are softer. Okay, it wasn't as big as we thought, but where we did see the biggest differences is after we applied our LAT-A to disrupt the cytoskeleton, what we see is that the cells take longer to recover from being compressed than if there was no, versus the control where no drug was applied to them. And with that, what we then realized really was that Mechano-NPS can measure slight perturbations to the cytoskeleton. But really what we find is that we're measuring the whole cell. We're measuring the nucleus. We're measuring many things with the cell, as it passes through our device. Now, what can Mechano-NPS really do? And here is sort of where I teamed with Mark Labarge, who used to be at LBNL, but now and has since moved to City of Hope. And what Mark's specialty is that he looks at breast cancer and aging. And in talking with him, we learned about the mammary duct and that there are 2 different cell types, the myoepithelial cells and the luminal epithelial cells. The myo-eps are responsible for ductile contraction, and the luminal epithelial cells are responsible for milk production. And they're more susceptible to being carcinogenic, to cancer, versus the myoepithelial cells. An interesting fact that we found from Mark in talking with him is that as a woman ages, the ratio of myoepithelial cells, or MEPs versus LEPs, changes. In young women, you have more MEPs than LEPs, and as women age, you start to lose those MEPs and you have more LEPs than MEPs. And that's the reason why women will, again, breast cancer is a disease of aging. Okay. And what Mark was wondering, once we were telling him about our Mechano-NPS device, was, well, if you can measure stiffnesses of cells, could you actually measure the differences between MEPs and LEPs? And so what he did was he actually sorted cells for us, and he gave us two vials. One was MEPs and one was LEPs of cells that came from a 66-year-old woman. And what he typically does is he stains these cells or tags these cells with particular probes that look for a particular marker on these cells, and then he sorts them. And it's quite a bit of process. And what we found was, yeah, we could measure them, but you can see there's a huge overlap if we look in our three-dimensional view. Once we take into account the size of the cell and look at WCDI, you can see that we like to say we got a signature. That is, we see that the LEPs are more soft than the MEPs, but yet again, you see that there's quite a bit of overlap. But nonetheless, the true test for us was whether or not we could measure MEPs and LEPs in a blinded sample where they weren't sorted, where they weren't separated from each other. And what we did was we basically used our data right here as signatures and then applied a maximization algorithm. And what you see here is data for young women from 19 years old to 29. The gray is our original data. This is WCDI on this axis right here. And essentially what we are able to do is pull out statistically the two different populations that were in our blinded samples. And this shows you the ratio of the MEPs to the LEPs. Shockingly, which it's still pretty neat, is the fact that biochemically, Mark was labeling these cells and was able to sort them, and that the ratios were identical to what we got, or we got the identical ratios of what he got, which again, this is a mechanical measurement that we're doing. Mark was doing a biochemical measurement, but we met in the middle in terms of being able to identify the different subpopulations. Here's what we have for older women, ages from 61 to 68. And again, you see in gray, that's our original data, and then what we pulled out statistically for the two subpopulations. Again, they agreed very well. So for us, at least we could say that we have agreement with a traditional biological assay. I think that one of the things that Mark was disappointed, and then I understood why, again, he's all about aging and breast cancer, and the hope was, well, would we see a difference in stiffness of these MEPs and LEPs as a woman progresses in age? And you can see that no, in fact, the WCDOs are roughly around the same, the average. But that's the best part of doing Mechano-NPS, is that you get multiple information out of your current pulses, and I knew that as an older person, I don't recover from injury like my graduate students who are far younger than I am, that in the end, what we should try to do is look at recovery times. And that's where we saw the biggest differences between young and old. So you can see here, so the lighter purple corresponds to recovery times, longer recovery times. So essentially, we couldn't measure any recovery from cells. And darker purple corresponds to immediate, that the cells just bounce back from being compressed. And you can see that in the end, older women's cells, HMECs, or these primary cells, breast tissue cells, essentially on average just don't recover as well as the younger women cells. So in the end, this mechanical measurement really enables us to detect chronological age difference, which really sounds like a really bold statement in many ways. And so the question that we really wanted to know was, in the end, is this recovery time, the differences that we're measuring between young and old, is it really young versus old? Or do we just luck out and that we just it depended upon the strains or the primary samples that we chose. So what we ended up doing was we did a lot of measurements, and I'm going to explain, but it turns out it really is a young versus old phenomena. And in addition, it's all about genetic risk factors, and I'll explain right now. So Mark has this amazing bio postdoc, Stefan Heinz, and he's the atypical biologist. He was totally into learning about new technologies. We actually set up, and I'll show in my final slide, how we set up our system in their lab. But he learned all about the technology, and what he ended up doing was measuring 19 different strains, or 19 different primary samples of different women, 19 women, and he essentially measured thousands of cells. And then what he did was essentially have this database of different parameters of each different cell, and then he essentially created a machine learning model, and he trained his model so that in the end he could classify cells based upon young versus old. And with that, what he was able to do here is a ROC curve, and what you see is that in the end, taking different strains or different primary samples, running it through his model based upon all the data that he collected for all the different cells, he can get an age prediction, again, of young versus old. His area under the curve is about 80%, which is really quite good. It turns out that you really do need all the information, the velocity of the cells, how much are they deforming and so forth, not just recovery time. And I liked how he plotted recovery time here to show that it really wasn't sufficient to be able to predict, well, young versus old. What he ended up doing as well, which is really, really quite beautiful, is that he ended up looking at high-risk strains. That is, those women who are young, but who have BRCA mutation and essentially are at higher risk for developing breast cancer. He ran it through the Mechano-NPS platform, collected all his data, ran it through his model, machine learning model, and did an age prediction. And this is the data that he sees. And what we find is mutation does matter. So here is just a young woman, a normal risk factor, and you can see the majority of the cells are behaving as young cells versus older cells. Younger women who have the BRCA1 or the BRCA2 mutation, what you can see is there's a larger fraction of cells that are acting as though they're old. That is, they're just not recovering from being compressed. So this is really quite astounding that, you can actually, this is something that Mark's group could not predict using traditional methods, but this is something that you could see just mechanically. And so the question is really why? Why are we able to see these cells essentially become act as though they're old if they have BRCA1 or BRCA2 mutation, or what's going on? And what we've sort of pinpointed in complement to some of Mark's other work, where they're looking at different filaments of the cytoskeleton, and in particular, a particular intermediate filament called cytokeratin 14. What Stefan was able to show was that if he silenced cytokeratin 14 and HMECs that were derived from an older woman, age 61 or 66, what he finds is that the cells spring back from being compressed much more than if the keratin 14 was there. What he also did was he took cells from a young woman, 19 years old, and essentially overexpressed keratin 14. And what you see here is that how much the cells, a greater portion of the cells dramatically do not recover from being compressed versus the control, where keratin 14 is not being expressed. And I think this is really amazing that you can actually see these differences and predict it just by doing mechanical measurements. In the end, what we feel like is that in the end, what we have is a chronological age of a woman, and then we also have a mechanical age of a woman. And our hypothesis is that the greater the difference between chronological and mechanical age really tells us the increase in the risk of developing breast cancer in a woman. We've also applied the same Mechano-NPS to look at leukemic blast cells. And for us, what we find is soft cells actually respond to treatment versus stiff cells don't. And that's another part of the story, and I'm sort of going long on my talk. I can talk about it for those who are interested later on. What I want to, we're really still building our story about mechanical states and breast cancer and what defines increase in risk. But what I wanted to just sort of show you is really how we're advancing Mechano-NPS. And I call it Mechano-NPS version 2.0. And in this case, what we're doing is we're measuring the viscoelastic properties. Again, all through this 4-terminal measurement, very simple measurement. Everything's run on batteries in the lab. And so with us, just as a reminder, with viscoelastic properties, we're interested in G prime, which corresponds to elasticity. G double prime corresponds to viscosity. And Jiangyang made this nice slide for me so that I would always remember what was G prime and G double prime. But essentially what we found is what we could do was, instead of having these nodes and pores and then this straight contraction channel, what we could do was we can actually make that contraction channel, instead of a straight one, into something that's sinusoidal. So as the cell passes through, it's being periodically deformed as it makes its way across from one end of the device to the other end. And then again, we apply our 4-terminal measurement. And what you see is sort of what the current pulses that we measure, that instead of a flat bottom curve in a straight contraction channel, what we get is something sinusoidal. Again, because the cell is deforming, it's blocking different current field lines, it's changing things, and so that's what we see in terms of the current. And with that, what we're able to do is we were able to look at 2 different cell lines. One was non-malignant, the MCF10As, MCF7s were malignant breast cells. And what we find is that we essentially get a power law behavior between viscosity and elasticity as we change the frequency of our device. And here frequency is we're changing the flow, the pressure of the cells passing through our device. Using this visco-NPS, as we call it, we can measure mechanical transitions as the cells undergo, for instance, the cell cycle. And so here what you see here is this is where we're looking at elasticity. It's very complicated, but nothing really dramatic. But here what we see is we see how the cell is changing based upon viscosity. And this is looking at cells as they go through the cell cycle phase, where they start at the beginning of the cell cycle. S phase is where DNA is being unwound from the histones inside the nucleus and being replicated. And G2 is where the cell is assembling everything back together and preparing itself for M phase, where the cells undergo mitosis and separate. Now you can see that there are differences, but the really cool part is that once you remove the cytoskeleton and really just look at what's happening in the nucleus and do this measurement, what you really see is this dramatic difference in the viscosity of the cells as they undergo S phase, as the DNA is being replicated. And essentially, again, what you can think of as is the cells unwinding the DNA. Essentially, it's becoming this blah, I like to call it, it's just becoming like this puddle of a nucleus. And then everything gets reassembled again and viscosity increases. So that was pretty neat. We also can look at something called epithelial mesenchymal transition, and this has a big role in cancer and wound healing, whereby cells are epithelial, they have certain properties. They like to adhere to one another, and they express different proteins, but then they go into a mesenchymal transition, they become more stem cell-like, they lose the ability of these proteins to adhere to one another, and they essentially become like these free cells in a tumor and get released into the bloodstream. We were interested in whether or not we could see differences there because everything is changing inside the cell. And indeed, we were able to induce EMT in cells using slug, and that's something a biological term. But essentially where we saw the biggest differences really was the fact that you could see, again, the viscosity changing as the cell undergoes EMT, and the viscosity increases greatly. And I should say, for those of you in the audience who do AFM measurements and have looked at EMT, you may say, wait a second, this is opposite of the effect that you see with an AFM. You would see a lower viscosity with cells that are undergoing EMT. And therein lies the difference between our device and AFM in the fact that what we're doing is measuring a cell that's in suspension, and we're measuring that whole cell changes. Whereas with an AFM, the cell is adhered to a substrate and it takes on some of the mechanical properties of that substrate. So there are big differences between the two different types of measurements. All this was bulk cell measurements. So what about single cell measurements? And this is very new. I have a new graduate student, bioengineering graduate student, and what he's done is basically concatenate a whole bunch of different sinusoidal devices together and doing multiple 4-terminal measurements depending upon, as we call it, the zone that we're looking for. And you can see that the sinusoid changes, we're increasing frequency as the cell goes from left to right. And with that, it's really neat that what you see are these different current pulses. Here he's flipped his current pulse, sorry about that. But essentially you can see as the cell is passing through, you can see that we're getting different current pulses. And this is raw data. We're seeing different sinusoids depending upon the frequency and the periods within each zone. And it is really cool that you see ripple effects of the cell even passing through zone 1. It has a downstream effect in the later parts of the device. We're still trying to figure out how to process this data and what it all means. So that'll be a talk probably next year. But again, we're still learning how to process this data, but I think what's really cool about this technique really comes from my background in physics and doing measurements off of wires and Josephson junction, was really how powerful this 4-terminal measurement can be. And if we go back to looking at this plot that I had for methods to perform mechanical phenotyping, what I can say is that Mechano-NPS, we're somewhere around here, maybe a little bit too ambitious saying our throughput is comparable to microfluidic cytometry, but nonetheless, we get a lot of resolution, and in particular, looking at recovery times. So just sort of wanted to end and just really show you what our whole platform looks like. It's very simple, and this was really a true, it was a really learning experience for all of us involved, in that we had to make a platform that was essentially like a black box, and we delivered that to the Labarge lab down at the City of Hope. You can see how very simple it is. Batteries, current preamp, a voltage supply in this case, and a computer. Now at this stage, what we do is we make devices in my lab and then we ship them down to the Labarge group. So it's a really neat collaboration that we have. And once a year we go down and we tweak the system for them and provide new software to analyze their data. So with that, again, many thanks for letting me talk about the project that we have going on in our lab. And I'd just like to acknowledge a great group of students. Those in gold are all the people who work on NPS past. Jung-Hyun Kim, he started a revolution in our lab. And the newest student is Rachel Rex. And all my collaborators who are either here at Berkeley or at the City of Hope, and my funding sources. Thank you very much.
Well, thank you, Lydia, for this absolutely brilliant talk. And it's given us a whole new perspective, I think, on mechanical engineering. Thank you for making it so clear. And it's quite different from anything else I've ever heard before.
Oh, thank you very much.
So let's see, we have a question here. From Jake Simon. Given that cancer cells are typically softer than healthy cells, why, from what I've heard, the tumors often feel harder than the surrounding tissue?
Right, that's a great question. So when you talk about a tumor, you're really talking about its entire environment. So that's including other cells, and it's including the extracellular matrix and all the different tissue around it, and that becomes much more. It turns out the stiffness is harder. But when you look at individual cells, if you think about their, especially in particular the invasive cells, they want to invade. They have to have that property to squeeze through extracellular matrix to make it into the bloodstream and so forth. So they are softer in general.
Thank you. So another one, amazing talk. Have you considered measuring other non-cellular things using this technique, other soft materials?
You definitely could measure different non-cellular materials, that's for sure. We've pretty much primarily stuck with cells, but there's no reason, as long as you have essentially a particle, we can measure it. That's what I tell everyone, so yeah.
So one thing I was curious about, you have mentioned early on that forensic breast cancer had now become the most common kind of cancer, whereas it used to be lung cancer. Has it that the rate of breast cancer has gone up, or that lung cancer has gone down, possibly because of less smoking?
Yeah, I think it's mostly the latter, that the incidence of lung cancer has gone down because of preventive measures, because of the smoking and so forth. I think breast cancer has pretty much stayed constant, but it's still amazing that it took over. And yeah, it shouldn't be. I think we all should be expecting things to go down and everything.
Someone says, great talk, I have to run, sorry. Okay, whoops, sorry, So another question, cell subjective to dynamical squeezing is out of its equilibrium state. Can you evaluate what the different mechanical values of cell in steady state and cell in out of equilibrium state?
I think you should be, if I understand the question correctly. So we've talked about, like, depending upon how much the cell is under stress, you can look at, again, different strains, but also how long that channel is, the contraction channel. How long it's undergoing that strain being applied. That's going to vary quite a bit. We've never really pushed it. But at the beginning, when we were trying to decide what was a sufficient amount of time for a cell to be under strain to get meaningful results, we settled on a certain time scale or length scale to do that. But I see that there's no reason why you shouldn't be able to do that.
And then we have another question from Ed Strez. I'm interested in seeing a graph of the ratio on the hesitance changes of experimental versus theoretical.
Wait, one more time.
I don't quite understand it. It says, I'd be interested in seeing a graph of the ratio on the hesitance changes of experimental versus theoretical.
I'm not quite sure, but what we're presenting, as had been with many of these, for instance, even the results that I showed of, like, Dubois and Bean results, a lot of that has been phenomenological. That is, you measure and then you start to fit everything in, and I think that's a similar case. As I showed you, we've done side-by-side experiments with micropipette aspiration, but actually doing sort of a theoretical model, we're trying to do that right now with our visco-NPS. The other ones are a little bit harder to do. And again, because we're measuring something that's different, cells are in suspension versus being adhered to a substrate. If they're adherent cells, it's hard to compare. You're comparing, I would say you're comparing a Fuji apple to a Macintosh apple, right? It's not quite apples and oranges, but it is different still.
Okay, the question got retyped. And
okay.
I'd be interested in seeing a graph of the ratio of the resistance changes of experimental.
Oh, yeah. That would take me a while to find that. Okay, I'm gonna get out of this mode and show. Hopefully I didn't do something. I'll bring out some old data here if you bear with me a moment. But yeah, I mean, I can, you know what, I can direct you to our paper that we have. It was one of our original papers, and it is in Review of Scientific Instruments in 2001, where we actually compared our results using polystyrene balls or colloids of different sizes and showed that we can see that crossover from the Gregg and Steidley model of looking where a particle is comparable to the size of the channel and where it's much smaller. We actually can measure that, see that crossover. And our results are actually quite nice. I'm sorry, it would just take a while, and I think given the time limit, and I'm also happy to converse later on in an email.
Thank you. So maybe you could go back into your original mode.
Oh, okay, I try. Oops, let me first do this presenter review. Sorry. Oh, okay, just a second. I'll just do it that way. Yes, can you see it now, my screen?
No.
No? Okay, just one moment. Play from current slide. Okay. Oh, I see what I'm supposed to do. Oh. Uh-oh. Can you see that?
No, I've seen.
Oh, wait. Oh, I have to do share. That would help. Now, can you see it?
No, I seem to have lost everything.
Oh, that's odd. Okay. One more time. Apologies. This is when it all falls apart, right? Can you see it now?
Ah, yes.
Okay.
But I seem to have lost
Uh-oh.
the chat and Q&A information.
Oh, let's see. Oh, I see. Let's see. Oh. There's one from Mike Deweese. To what extent does lowering or raising of electrical current around a cell reflect changes in orientation of a non-spherical cell in the channel? So we actually do see, so the presumption is always that the cell is spherical. And in that case, in the model, you should see a flat bottom curve. And we see that for the most part. But we've actually taken cells that were non-spherical, and you can actually see that in the shape of the current pulse at the current pulse bottom, the shape of it. And this was actually done by, it was back in GE days when, and they were looking at different colloids that were either dumbbell shaped or not, and as they pass through a pore, and you can see the shape of whatever passes through. Again, this whole idea of that you're affecting the electric field lines in the channel. So you can actually measure shapes of cells in the device. And also, does the current flow around the cell give info on the shape? Yeah, beyond its cross section. It does, and this is some of the very old work that we've done where we actually started to take colloids, and then we started to do a sandwich assay. We started to put different molecules on the surface, and the colloid surface would be on a molecular scale, sort of rough, if you can imagine. I hate to use the analysis of, I'm not going to do it. I was going to say it's like the COVID virus for SARS-CoV-2, where you have spikes. Ours would look molecularly like spikes, and you can actually see that shape and extract it out of our current pulse. It's actually quite interesting because some current pulses are, some electric field lines are compressed, and that changes the current measurement that you measure and so forth. I mean, yes, in the end, I'm being long-winded, but yes, you can see surface changes, different shapes. So,
yeah, Mike DeVries says, thanks, really interesting talk.
Yeah, thank you.
Another maybe somewhat obvious question is,
Mhm.
you've done this, made this enormous effort to study breast cancer, but what about prostate cancer and other kinds of cancer? I mean, do you think you can use the same techniques?
Oh, absolutely. Absolutely. You could use the same technique. Yeah. You could do it with prostate cancer. We've talked about, actually also even outside of cancer, you can use stem cells and so forth as well. It's a general way to characterize cells in general.
Okay, any other questions? Well, Lydia, thank you once again. This is a whole new perspective on looking at cancer that I'd certainly never come across before. And as I said, I have another view now of what people do with mechanical engineering.
Well, thanks very much. It was really nice to catch up with everyone and talk in the department.
Yes,
I appreciate it. It was an honor. So.
And I know we can't give you any applause, but great your effort and your energy in this talk, and Lydia. So
thank you so much.
I know you're off to another part of the country.
In a few hours, yes. So.
Yeah. Okay.
Okay.
Bye.
Thanks very much. Take care. Bye.